Okay, so we're going to talk about GH. GH is all about GLSs. If you can do the GLSs, you can probably HD it. Again, we're going to start off with the genes. So this is just a sequence of DNA. Uh, Lots of high school biology here, transcription, translation, how proteins are made. I'm not going to get into it. Maybe a YouTube video will help if you don't have this base knowledge, but I'm assuming that a lot of people do. So you're starting from a gene, you're going to the mRNA via transcription, mRNA to protein via translation, and those proteins can then create protein complexes that are used all throughout the body for a multitude and plethora of reasons. So all our genes are going to essentially build our genome, and essentially all of our mRNA and transcription is going to build our transcriptome. So this is all our transcripts. And our proteome is going to be essentially all of our proteins and the different combinations that they make. Now, in terms of our base pairs, A goes with T, C goes with G. I just write it as ATCG. Therefore, if you memorize it as writing ATCG, you don't have to remember which base goes with which one. And obviously, the A and U also happens. Uracil replaces thymine later on, as per high school biology. I'm sure we all know that. So adenosine plus thymide is going to be these two bases here, and cytosine and guanine is going to be our C and G. And interestingly, there actually is no connection between organism complexity and our genome size. So the number of genes we have actually doesn't really correlate with complexity at all. Rather, there are these segments called non-coding segments. So essentially, these don't really code for anything, or we don't really know what they do. But we have noticed that these actually correlate with development complexity. So people who have more non-coding segments, or organisms who have more non-coding segments, usually are more complex, likely has some evolutionary reason. However, I do not know. That might be a question for the lecturer. So in terms of our maternal ancestry, this can be traced back to Africa with mtDNA. And interestingly, people in Europe came from a combination or from one of 7 ancestors. However, we can all trace it back to one common ancestor. I think it was like 150,000 years ago from my memory. Now, sequencing the Y chromosome also can track back our male ancestry. However, we've noticed that there's minimal variation here compared to the maternal ancestry. Now, the Genome Project was this legendary project where they sequenced the human genome. This project actually set up the groundwork for many of the advancements in gene technology that we have today. So this project is very, very important. Francis Collins is the person who did the publicly funded project, and then Craig Venter is the one who did the private project. And they both had pros and cons for as to why they wanted public or private. I think there was like a documentary in the GLS. Really interesting. If you have time, I'd really watch it. I personally really enjoyed it. Then you're going to have your chromosomes. So chromosomes, again, just a high school biology review. They're just compacted pieces of DNA. They have multiple genes in them. And this is some new nomenclature that we do need to know for this semester. Very important. So you're going to start off with the chromosome, whatever it may be. Then you're going to have 2 arms, P or Q arm. Now, the Q arm in a chromosome is always going to be longer than the P arm. Even with our metacentric chromosomes, where the centromere is in the middle, and it seems as if both are the equal length, both arms are equal length, there's always a longer arm. So there's always going to be a P arm. There's always going to be a Q arm. So you write P or Q. Then we put the band number here and subband. And band number is basically just going to tell us which where it is, and the subband is even more detailed description. Don't worry about the band and subband. The main idea is that chromosome, long or short arm, and then band and subband. So that's the main thing for that. Now you have 3 types, and yes, you actually need to know which chromosomes out of the 23 that we have are going to be acrocentric, which out of the 23 is submetacentric, and which out of the 23 are going to be metacentric. And I think this is like a week 2 or week 3 thing as well. In terms of our acrocentric, all this means is that our P arm is really short, almost to the point where it doesn't really exist, but it does. And then we have submetacentric, so our P arm is distinctly shorter than our Q arm. And then metacentric, it seems as if our P and Q arms are about equal distance, but obviously one is still longer than the other. Now, in terms of looking at a more detailed view, these tips at the end are called telomeres, and then you're going to have something called heterochromatin and euchromatin. So heterochromatin is going to be very dark, and essentially it's going to be silent and more condensed, and euchromatin is going to be light and relaxed and active. This is how I like to think of it. However, pathophys wise or anatomical wise, however you want to call it, I might be wrong, but I'm just trying to explain a point. My point is that if there's less darkness here, there's more space for binding. Therefore, it is more active. So the euchromatin would be more active in that regard. Because heterochromatin is very tightly compact, maybe it's harder to have binding, harder to have transcription translation, because there's less space for the different molecules to go in and work their magic. Hence, it's silent and more condensed. Heterochromatin, I like to think of it as hetero being different. Therefore, if we have all these different things mixed up together, or if you mix up a couple different colors together, you end up always getting the color black or a really dark color. So that's how I remember heterochromatin as being darker. Now, in terms of karyotyping, all karyotyping is doing is it's showing us for that one individual that we have karyotyped what the different chromosomes are going to look like, and our autosomal chromosomes are going to be from 1 to 22, and our sex chromosomes are going to be 23. Obviously, if you have an XY, you're a biological male, and if you have an XX, you're a biological female. However, there also are variations to that, so I'd like to acknowledge that as well. And our autosomal chromosomes are usually just the things that are making up the chromosomes for our body. So I guess that's what that means.